Hello everyone. Welcome to the second lecture of the 8th module, which is on neuromorphic computing and neurobiological concepts. So in the last lecture, we were discussing about the different parts of neurons. We saw how neurons generate that action potential or spike and how that action potential travels to the axon and reaches the synapse. And in the synapse, what happens is it changes the membrane potential, leads to opening up of ion channels, and then because of those opening up of ion channels, some of the calcium ions enter the vesicle. They kind of open up the proteins which hold the neurotransmitters. And then those neurotransmitters kind of diffuse through the cleft and move on to the receptor sites on the dendrites of the other neuron or the postsynaptic neuron. And the moment these neurotransmitters bind with those receptor sites, what happens is they kind of polarize or hyperpolarize, and in they depolarize or hyperpolarize the postsynaptic neurons membrane as well, and that leads to a change in the membrane potential of the postsynaptic neuron. So that is what we called as a PSP, or a postsynaptic potential change. And even the strength of the change in the PSP is determined by the synapse. So what a synapse can do is synapse can lead to different number of receptors which are binding to a different number of receptor sites or different number of neurotransmitters which are being bind to the receptor sites. And it can also leads to a change in the speed of their traversal. So due to all these properties, a synapse can also change the intensity by which the membrane potential of the postsynaptic membrane changes. So that is known as postsynaptic plasticity, or I would say this is known as synaptic plasticity. So let us talk about this synaptic plasticity in more details. So the strength of the synapse is kind of determined by the intensity of change in the PSP that it leads to, because the spikes which are coming out of the presynaptic neuron are the same. So if for a particular value of spike, there's a large change in the PSP, then this simply means that the strength of synapse is more. And that is what is defined by your synaptic plasticity. So what exactly is synaptic plasticity? It's the ability of your synapse to change its strength in response to the neuronal stimuli. And this stimuli not only depends on the presynaptic neuron, but it actually depends on the difference between the spiking pattern of the presynaptic neuron and the postsynaptic neuron. And I'll talk about that later in the next slide. So if somehow the synapse kind of increases in strength because of the neuronal stimuli, then it's called synaptic potentiation. And similarly, if the synaptic strength kind of decreases, then it's called synaptic depression. Now, these kind of synaptic plasticity behavior or these changes can be either short term, which lasts for some seconds or some minutes, or they can also be a long term kind of change in the plasticity, which can be ranging from hours to some days to some years even. And mostly it's the long term synaptic plasticity which is responsible for all the learning and the memory that we have. So our whole intelligence is kind of depending upon these long-term synaptic plasticity. So if the strength of these synapses had to remain same, we won't learn anything at all. It's only because of this ability to change the strength of the synapse that we learn new things. And it's actually, the synaptic plasticity is nothing, but it is related to the flow of calcium ions. So if there's a change in the number of neurotransmitters, or the response of those receptor sites, that is how you change your synaptic plasticity. I mean, if you increase the number of neurotransmitters, then definitely you'll increase the potentiation, then definitely you'll increase the strength of the synapse. Similarly, if you increase the response of those receptor sites, you'll increase the strength of the synapse. And vice versa, if you decrease all of them, then you kind of decrease the strength and you lead to synaptic depression. Now, what this LTP does is it improves the postsynaptic sensitivity to the neuronal signals that it is receiving from the presynaptic neurons. So if this strength is increased, then your postsynaptic neuron will kind of be more sensitive, or it will kind of process the information that it receives from the presynaptic neurons in a much better way, since it's amplified by the synapse that is connecting this presynaptic neuron to the postsynaptic neuron. And similarly, the opposite happens in the long-term synaptic depression. So what happens there is the sensitivity of the postsynaptic neuron to the signals received from presynaptic neuron is kind of decreased. And the best part about this plasticity is that it's kind of bidirectional, and mostly it's continuous, and you receive or you have like analog levels of synaptic conductance. So there is a continuous range of synaptic conductance to which you can kind of tune your synapse to with the help of your neuronal stimuli. And that neuronal stimuli, as I already told, that actually depends upon the spike timing difference between the postsynaptic neuron and the presynaptic neuron. And that is governed by a learning rule, which is known as STDP, that I'll discuss in the next slide, as I already told you. However, some synapses, like there are some neurological studies which show that some synapses show a discrete, more digital-like conductance as well. So for some synapses, the change in the conductance is not continuous, but it follows, like the conductance can take some discrete values. So it's like similar to the neurons, like some neurons show you action potential kind of a behavior, some neurons act like a frequency kind of processor. So here also some synapses show you kind of a continuous plasticity, whereas some of the synapses show a discrete kind of plasticity. And what is the reason for this? So the biological reason for this is the change in the quantity of neurotransmitters or the increase in the sensitivity or response of the receptor sites. That is how you can change or you can increase or decrease your synaptic plasticity or synaptic strength. Now, how this is modeled? So synapse is nothing but a variable programmable resistor. I mean, electrically, like your neurons were modeled as a capacitor with a leaky component or with some conductances as well. Similar to that, a synaptic or a synapse is kind of modeled as a variable programmable resistor. So it's like a two-terminal device where just by the action of different spiking pattern on the two ends of its this resistor, you can change its conductance. So that is why I told that it's modeled as a variable programmable resistor. Now there's a concept there's a very unique concept from neurobiology. So you must have read that practice makes a man perfect. So is it only a quotation or does it have any scientific significance? So the thing is, each task that you perform, there's a different set of neurons which are connected. So let me give you an analogy. So usually it's called that practice makes a man perfect. So is that restricted to quotations itself, or does that have any physical meaning, or I would say, does that have any scientific backing as well? So the thing is, yes, it is actually true. Practice does make a man perfect. Why so? Because, you know, for performing each of the tasks, there is a particular combination of neuron and synapses, which are there are a particular combination of neurons which get connected in order to perform a particular task. So if there are like hundreds of tasks that you perform throughout a day, there are 100 different sections of neurons which are kind of connected for performing each of those tasks. So if you perform one task more number of times, then what happens is the strength of the synapses that connect those neurons, that kind of increases. With every time, with every attempt to that task, the synaptic plasticity, or I would say the synaptic strength to the neurons which are responsible for that task, with that increases. And because of that, we say that the more number of times you do it, the better your performance becomes. And this is something which is also responsible for your involuntary actions. For instance, I would just give you a simple example from cricket, that the moment you see a ball coming at a particular location, there's a judgment that comes inherently from your side, right? As a batsman, that judgment comes inherently from your side, and you play a particular shot, depending upon the location of the ball that you are seeing. So how that judgment becomes perfect? That judgment becomes perfect only when you practice that shot a lot number of times. Only if you see the ball a lot number of times and you practice that shot a lot number of times. So that is what people say when they mean that practice makes a man perfect, and it has got a scientific backing. Basically relates to this synaptic plasticity. So whenever someone says you that, okay, practice makes a man perfect, next time you explain to him why exactly it makes sense. Now let us talk about the learning rule, which kind of enables this synaptic plasticity, or the learning rule which kind of helps you decide what is the exact value of the conductance or the strength of synapse, which is perfect for this kind of application. So one of those learning rule, which is also the simplest kind of learning rule, is called spike timing dependent plasticity, or STDP rule. So it is the process or the learning rule which kind of adjusts the efficiency or the strength of the synapses based on the relative timing of the spiking of the pre and post synaptic neurons. So it's also following the Hebb's rule. So what this Hebb's rule says is neurons that fire together wire together. Now what that means is, if you're firing if the firing difference between the presynaptic neuron and the postsynaptic neuron is very small, I mean, if the presynaptic neuron fires and immediately after that the postsynaptic neuron fires, then we can say with some amount of certainty that the postsynaptic neuron was kind of more sensitive to this presynaptic neuron, and let us then increase the synaptic strength which connects these two neurons. So what this STDP rule says is, if the postsynaptic neuron fires before the presynaptic neuron, that is, if the postsynaptic neuron fires and after that the presynaptic neuron is firing, then there's a high chance that there is no correlation between the two. There is no actually, there is no kind of relation between the two. There is no correlation between the two, and as such, since there is no correlation, the synaptic strength can be reduced. So what this learning rule says is, the moment a postsynaptic neuron fires before the presynaptic neuron fires, we kind of perform this long-term depression, and we weaken this connection between the two neurons. Similarly, if postsynaptic neuron fires immediately after the presynaptic neuron, it simply means that there's a high possibility that the stimuli from the presynaptic neuron led to this change in the membrane potential of the postsynaptic neuron, and it fired. So in that case, what should we do? We should increase the strength, or we should strengthen the connection between the two neurons, and this process is called long-term potentiation. I mean, we already know that this is kind of a part of plasticity. So this is the learning rule. And also, usually there is this antisymmetric STDP rule which occurs in the neurons of hippocampus. So I'll let you know what exactly it is by showing the diagram. So plot on your right simply explains the change in your postsynaptic potential as a function of the spike timing difference between the presynaptic neuron and the postsynaptic neuron. So this x-axis is nothing but a delta t, where this delta t represents t pre minus t post. That is the difference in the spiking timing of presynaptic neuron and the postsynaptic neuron. What this means is, if delta t is positive, so if the presynaptic neuron fired before postsynaptic neuron, so if the presynaptic neuron fires before postsynaptic neuron, then what we do? If pre is firing before post, then it simply means that the post firing kind of depends exclusively on pre. I mean, there's a high possibility that the postsynaptic neuron fired because of this presynaptic neuron firing. So in that case, we increase the potential, we increase the strength of the synapse. We perform potentiation, LTP. So this is what it shows you. So whenever your delta t is positive, it's more than 0, we kind of increase the synaptic strength, which is done over here. And similarly, if the post fired before pre, in that case, there's no correlation between the two, and it is highly unlikely that the post fired because of this pre. So in that case, we reduce the synaptic strength, we perform a depression, and that is what it has done. So it has reduced the synaptic strength. And also, if you see that if the difference between the post and pre firing instances is small, then the magnitude of change is large. That is just to ensure that the smaller the difference between the two, the smaller the difference between the pre and post firing instances, it simply means that there's a larger correlation between the two. There's a larger dependence between the two. And just to capture that impact, they have kind of this learning rule kind of has this kind of a dependence. And this is usually called an antisymmetric exponential STDP rule. And this is kind of very common in the neurons of our hippocampus. So this is the STDP rule simplified for you. So simply said, if pre fires before post, you do LTP. If pre fires after post, then you do LTD. And hence this is justified that neurons that fire together wire together. I mean, if pre fired immediately before post, then they have to be wired together. I mean, the strength of the connection between the two has to be increased. LTP has to be performed. So that is the major idea behind this. Now let us go ahead and read about a simple visual processing system, that is retina. So in our retina, what do we have? So a retina kind of converts the light to spike trains. So light comes in the form of photons, and this is converted to spike train, and then these traverse through our optic nerves, which feed them to the visual cortex, where they are processed. So what happens in your retina is that first the light stimulus, that is photons, they're converted into electric signals by the help of these photoreceptors. So what are these photoreceptors? So these photoreceptors are nothing but rods and cones. So you must have read about rods and cones in your 10th class. So these rods are nothing but they are sensitive to the intensity of light, and these cones are nothing but they are sensitive to the color of the light. So you may know that these hens have only rods and not cones. So they are colorblind, but they can their rods are so strong that they understand when the morning has happened, and they kind of indicate by a bang that okay, morning is there. So something like that. I mean, let us not go into detail about that, but yeah, that's the function of rods and cones. So this is how your retina looks like. So you have like rods and cone receptors, and then you have this intermediate layer of neurons. So there are some horizontal neurons, bipolar neurons, and amacrine neurons. So let us discuss what these immediate neurons do. So first, the light is converted into electrical signals by these photoreceptors, and then these intermediate neurons, which are bipolar, horizontal, amacrine cells, they combine the signals from several photoreceptors. So there are like 100 million photoreceptors, and the number of kind of these ganglion cells, which are like the further level of this retina system, is only 1 million. So these intermediate neurons kind of combine signals from 100 million of these photoreceptors, and they kind of compress it to 1 million informations. So it's like, so these neurons kind of act like a signal compression system, and they compress it by a factor of 100. And once they compress this signal from all these photoreceptors, they send it to the ganglion cells. So there is this retinal ganglion cell which form the last portion of the retina, and then these ganglion cells are kind of responsible for generation of spike. So these ganglion cells generate spike train based on the spatial and temporal patterns of the information or the electric signals that they receive via the photoreceptors and the intermediate neurons. And then the action of this ganglion cell is kind of connected to the optic nerve, and this conducts spike trains to the cerebral cortex or the visual cortex through LGN of the thalamus. So I won't get into details of pure biological stuff. But now what happens is your action of ganglion cell is kind of connected to the optic nerve, and it conducts signal to your visual cortex. Now, how does a visual cortex process this signal? So visual cortex has several layers of neurons, and these are arranged in the form of hierarchical form. So the information traverses in a hierarchical manner. So information is fed into the first layer of neurons, and then that information goes to the second layer of neurons, then third layer of neurons, and there's no feedback here. So this kind of propagation is called feed-forward propagation. If there's a feedback, it's called a back propagation, which is very common in your neural network training and all, and that we'll discuss in the next lecture. So anyways, so here the information traverses in the hierarchical manner in a feed-forward way, so that if there are different layers of neurons in the visual cortex, then information will flow from one neuron to the next level of neuron, and so on. So we have the first layer of neuron, which is called the V1 layer of neuron. So all these neurons act like a spatio-temporal filter or a Gabor filter. So they kind of extract some patterns, the patterns depending upon the location, which is the spatial information of the light, and temporal, that is how these light intensity changes with time. So these two informations are kind of extracted by this V1 layer of neurons, which are kind of modeled as Gabor filters. Then there's information propagates to the V2 layer of neurons, or the next higher layer of neurons, and there they extract finer features such as the shape of the object from which this light is being emitted, the edge detection, then surface segmentation, I mean the surface of the object from where the light is being emitted or which is reflecting the light, and even the direction. So these finer features are kind of extracted by the layer of neuron which is after the first layer. And if you talk about the other layers that are immediately after it, then they will extract some other finer features or some other advanced features. So this is the kind of hierarchy that we have in any neural network. So the first layer of neurons will kind of take in the input. The second layer will extract some features from it. The third layer will kind of extract some more features. And as you go along the layers, I mean, as you go on top of this hierarchy, what you will see is that the number of features that are extracted, their quality improves. So that is why it is always advantageous to use many hidden layers in between the input and output layer. So this is also something which will become clear as we study the next lecture. So, okay, we know now about the V1 layer of neuron, V2 layer of neurons, what are the features that they extract. And the best part about these layers is that these neurons learn in an unsupervised manner, utilizing your STDP rule. So it's not like you're training it by showing it some data and then kind of checking whether the output of these neurons is same as the data that you showed it to or not. But these neurons learn in an unsupervised way, in an autonomous manner. So this is the best part about this kind of retinal, like this retina visual processing system. I'll share a fun fact with you. So what scientists did as a part of an experiment, that they identified first the visual cortex of a rat's brain. That is the portion of the brain of rat which was responsible for vision, where these retinal ganglion cells were sending information. And then what they did was, instead of, they just cut down those kind of nerves, optic nerves basically, and then they artificially connected the cochlear ganglion cells. So the cochlear ganglion cells are kind of responsible for hearing. So they kind of connected its hearing capability, or the cochlear ganglion cells, to this visual cortex, and then left it to train on autonomous way, or left it to train in an unsupervised manner. So what actually happened was that the synapses of this visual cortex actually trained in an unsupervised way, and the rat was able to hear even though this visual cortex was responsible for vision. I mean, this visual cortex, if connected to the cochlear ganglion cell, just because of its capability of unsupervised learning, can even enable hearing. So this was one of the experiments which demonstrated that this unsupervised learning, with the help of this unsupervised learning, the synapses can learn almost anything, if only if they are left, or if they are allowed to train for a long period of time using this unsupervised learning rule itself. So that was one of the major breakthroughs, or I would say that was one of the very highly conclusive experiment that scientists performed on this rat. So we discussed a lot of biology till this time. Let us go on to VLSI. Let us explore how nanotechnology or VLSI can be used to realize all these systems. So let us start with hardware implementation of synapses. Surprise, in 1960 itself, people came up with this memristor synapse. So this is memristor, not a memristor, which was discovered way in 2000. So this memristor synapse was kind of fabricated way back in 1960, and it was also used in a system which was called Adeline neural architecture. So this memristor is not a two-terminal device like a memristor, but it's a three-terminal device. And what exactly is this memristor? So this memristor actually works on the principle of electroplating. So before we get into the electroplating and how a memristor works and all, let us discuss something about this memristor, which are more popular because it's two-terminal architecture. So memristors are two-terminal, and they were conceptualized by a professor of UC Berkeley, Leon Chua, in 1971. So what did Chua see? So Chua found out that there were some elements relating these four kind of properties. So charge was related to voltage by a capacitor, voltage was related to a current by resistor, and even current was related to your flux with the help of this inductor. But there was nothing which could relate your flux to charge. So based on this kind of an intuition, Leon Chua proposed that there would be something which is called memristor, which would kind of relate the flux with the charge. And this d phi equals to m dq would be the kind of relationship between charge and flux given by that memristor. But are RRAM memristors, are PCM devices memristors, or whatever we call memristors, are those really memristors? The answer is no, because there's no flux there. There's no magnetic flux there, and there's no relation between flux and charge that your RRAM or something like PCM device will give you. But why they're called memristors? Just because they were two-terminal devices, and even the definition of memristor was kind of and even the definition of this memristor was kind of relaxed by Leon Chua when he saw the 2008 paper on the device which could show a change in the conductance as a function of the applied pulse across it. So that is how even those devices started being called memristor, but they're not actually fulfilling any of these conditions. So immediately after that paper was published, missing memristor found, there was another paper which came in Nature itself, and that paper's title was Missing Memristor Not Found. And there was a huge round of discussion between the authors, and it is very interesting to read that. I would suggest you guys to read it. So now let us come back to memristor. So memristor, as I told, is based on a reversible electrochemical reaction. So it's a three-terminal device. So what happens exactly here is, so this is the test tube in which you fabricate your memristor. And in this test tube, you fill it with a solution of copper sulfate and H2SO4. And then you dip a copper rod in this solution. And there's this, you dip a graphite rod on this solution. And then you have a copper wire, which also kind of, is dipped inside the solution. So what happens is, depending upon the current that flows through the copper wire, there is this electroplating of copper onto this graphite rod. And that electroplating of copper from this solution onto the graphite rod, that actually changes the resistance of the graphite rod. And that is how they kind of implemented a synapse. So this is how people first came up in 1960 itself with the concept of a synapse. So apart from memisters, as I already discussed, that even the EEPROM cells from your Intel Etan chip, I told that they used 10,240 synapses, which were floating gate based. So these were kind of EEPROM cells. So even floating gate cells were used in the form of EEPROM for synaptic application. However, in those days, the synaptic application was not based on the flash cells exclusively. So what people used was they kind of used these flash cells in a differential amplifier kind of architecture. So what they did was they tuned the threshold voltage so as to tune this del VFG. I mean, the difference in the threshold voltage of these two EEPROM cells could be tuned by changing their threshold voltage, and then the effective I out or I plus minus I minus of this differential amplifier kind of depends upon this del V in that we are applying externally, as well as this del VFG, which we are obtaining by tuning the threshold voltages of the two EEPROM cells. So it would be a good exercise to show that this I out, that is I plus minus I minus, is actually proportional to GM del V in times del VFG. That is an exercise that you can do independently. So why differential amplifier? Because this would increase the kind of dynamic range of the current. This would increase the del I that you could get. In those times, the synapses did not have this kind of a tunable capability, and even the oxide thicknesses were too large, and the kind of what should I say, the kind of voltages that they used to operate with was also very large. So they used a differential amplifier to increase the range and to make it more sensitive to this change in the threshold voltage. That was the reason. Problems with this kind of structure or any floating gate, even now, it's a 3-terminal devices, while the biological synapse is only 2-terminal. Also, the operating voltage is too high, even for logic, even compared to the logic operations, even now it's too high, and the endurance is limited, right? So you have only 10^5 cycles of change in the synaptic plasticity, whereas you need a lot more than that for any practical, viable, or any useful neural network application. Then people also came up with this DRAM-based synapse. So what they used was they kind of put this 1T 1C DRAM as one of the input terminals of this differential amplifier. Why differential amplifier? Just to make this more sensitive to the change in the charges of the DRAM or the storage state of the DRAM. So that is what the concept is. You tune the del V in by changing the memory state or the charge on this capacitor. So your del I will depend on your V in, and it will be extremely sensitive to the change in the differential voltage. And the part is, you can implement STP with it. I mean, you can implement synaptic plasticity with it, but the only problem is you require constant refresh even here. So that is something which prevented the use of DRAM. Also, for implementing one synapse, you need here 10 transistors plus 1 capacitor. So the kind of area that you require for implementing one synapse would be very huge. So people also came up with this, SRAM-based implementations. So the SRAM-based implementations were not exactly 60 SRAM-based implementations, but they were modified SRAM-based implementations. One of the commonly used SRAM for this was this transposable 8T SRAM cell. So with SRAM cell, you could easily realize binary weights, right? We could easily tune the weights as 0 or 1. However, it's volatile. That's its biggest problem. And it requires large area just for storing 2 weights, 1 and 0. So that is one of the major problems. However, we can use NVSRAM that can kind of remove its volatile property, and that can make it non-volatile. But at the same time, you increase the number of memory elements or the transistors, and the area increases further. So how these emerging non-volatile memory-based implementations are more promising? First of all, they are 2-terminal. And second of all, let us first talk about PCM. So for your PCM, you increase the series of pulses in your set state. You kind of crystallize a larger amount of amorphous region, and you increase the resistance. Similarly, you decrease the magnitude of the pulses. Then what you do is you kind of melt increasing amount of that crystalline material, and you reduce the resistance of your material. So this is how you can achieve synaptic plasticity in your PCM device. Now let us talk about RRAMs or CBRAMs. Okay, so CBRAMs also similarly, you apply for CB-RAMs, the best part is even if you apply, or RMs also, the best part is even if you apply pulses of identical amplitude, then what happens is you achieve increase in your conductance or a decrease in your conductance. And why does that happen? So even if you're applying like same pulse every time, you're kind of providing some energy to your synapse, some energy to your vacancies and your ions, so that based on those energy that they receive, they can either recombine or they can then be attracted towards the electrode. So the best part about CB-RAMs and RRAMs is that they don't require this kind of a increasing amplitude kind of pulse for potentiation or depression. Even for RRAM, you can apply same same kind of pulse, and you can see that the conductance increases and similarly the conductance decreases. However, for PCM, you always require this kind of increasing pulse waveform or decreasing pulse waveform. For reset especially, if you apply same amplitude of things, I mean same amplitude pulse for resetting a PCM, as I told that you will be melting same amount of amorphous material, you'll be melting same amount of crystalline material and the resistance won't change. However, for set state, even if you apply same amount of same pulse with same amplitude, you can still achieve a potentiation, right? So what people did, or what exactly happens in neural networks? So your neurons are kind of providing you an identical pulse every time, right? So if the neurons are providing identical pulse, then we cannot have this kind of pulses with increasing amplitudes in your kind of networks. So we'll always have identical pulses coming at the two terminals of your synapses. And if at all potentiation or depression has to happen, it has to happen with those identical pulses itself. So to work with identical pulses in PCM, what they proposed was this kind of a differential kind of circuit. So here what they do is they kind of take two different rows. One is dedicated to LTP, one is dedicated to LTD. However, the thing is they are not doing LTD here. I mean, they are not using this kind of mechanism. So both of them are being programmed to set state itself. And this is the current through this is kind of subtracted from the first one. So if the set state of the first one or the current through the first one is higher than the current through the second one, then we say that it's potentiation. However, if we want to depress, what we do is we kind of reduce the resistance of the LTD part. That is, we set it to more lower level of resistances. And then what exactly happens is the current through this is larger than the current through the LTP module, and the overall current, which is the difference of the two, that becomes negative. Or we say that, okay, we have depressed our device. So just by operating in the set condition itself with identical number of pulses and utilizing this kind of a differential configuration where the effective current is the difference between the two, you can also realize a kind of potentiation as well as depression behavior using PCM in the set mode itself. However, one problem that is there with all the emerging non-volatile memories is variations. So you apply identical pulses, the conductance increases, okay. But at the same time, if you kind of plot the conductance of the different devices in that array, all of these conductance will be different. I mean, you see, there's a huge distribution of the conductance of the different devices. And that is not the case in biological synapses. So uniformity is something which is important. But at the same time, since biological synapses can work with some amount of these variations, I mean, the degree of variation is there in biological synapses also, but it's not this huge. But they can also work well with some amount of variation. And as such, this provides us some leverage to work with this emerging non-volatile memories, even for synapses. Now, once we have looked at the implementation of synapses, let us also look at the implementation of the STDP rule using your RM devices or PCM devices. Now, let us also look at the implementation of the STDP rule. So most of these implementations of the STDP rule based on emerging non-volatile memory is based on pulse shaping. So what they do is they kind of shape the spike in this form, and then they apply it to the synapse. I mean, the synapse here is RAM. So this is its top electrode. This is its bottom electrode. And what they say is that this line corresponds to the postsynaptic neuron. And this line, or the horizontal row, that is the top electrode, is being fed by the presynaptic neuron. And based on the difference of this spike signal, what actually changes is the voltage between the top electrode and the bottom electrode, and that kind of determines the conductance of your RM. So what happens usually is if your post spike is kind of arriving before the pre-spike, that is, you can see here that the post spike arrived before the pre-spike. So what this leads to is that the top electrode, so the top electrode of this RM is being fed by the presynaptic spike, and the bottom electrode is being fed by the postsynaptic spike. So the maximum difference or the maximum voltage difference between the top electrode and the bottom electrode comes at the instance when this spike has its the minimum voltage, and this postsynaptic spike is at its maximum voltage. So at this instance, what happens is maximum voltage drop occurs across this RM device. And what is the direction of that voltage? So here the postsynaptic spike is positive. So your bottom electrode is at positive voltage and your top electrode is at a negative voltage. So your top electrode is at a potential which is smaller than the bottom electrode, and the difference between the two is huge. So what this leads to is, this leads to an effective negative voltage appearing across this RM, and this leads to reset of this device. And reset of this device will lead to what? It will lead to an increase in the resistance or reduction in the conductance. So effectively what we want is, we want the conductance of the device or the strength of the synapse to reduce when the postsynaptic pulse arrives before the presynaptic pulse. So this is what exactly is happening for this RM. So what exactly is happening is, once this postsynaptic pulse is arriving before the presynaptic pulse, effectively this minus V reset voltage falls across this RM, and it kind of reduces its conductance or increases its resistance. So this is how STDP is implemented. Similarly, if the pre-spike comes before the post-spike, so here you can observe that the pre-spike has come before the post-spike. So what happens here is that since the pre-spike is kind of feeding the top electrode of this RM, so the maximum voltage drop across this RM occurs at this instance when the post-spike is at its minimum and the pre-spike is at its maximum. So in that case, what happens is, the effective voltage across this RM is, like on the top electrode, the effective voltage is positive. On the bottom electrode, the effective voltage is negative. So the maximum voltage that is occurring across this RM is in such a way that top electrode is at a higher potential as compared to the bottom electrode, and as such, set happens in case of this RM. And because of set, what happens? Your conductance increases or the resistance reduces, and that is precisely what we want. If pre is coming before this post, so if pre is coming before this post in the time domain, so if pre comes before this post, what happens is, in most cases, or I would say there's a high probability that the post was caused because of this pre, and as such we increase the synaptic strength or we increase the conductance or the strength of the synapse, and that is what we precisely observe here. And also this del T, as your del T increases, the amount of voltage that drops across this RM, that reduces, and as such the level of kind of change in the conductance kind of reduces as the delta T increases. So just by shaping this pulse, like this the post neuron and the pre-neuron synaptic pulses, we are able to achieve STDP in case of your emerging non-volatile based memories. So this can be RM, this can be PCM, this can be other devices as well. So we can discuss them at long stretch, but we don't have enough time. So this is the general idea how we can shape these pulses and kind of achieve STDP route. However, one very, I would say, an implication of this variation is that the set threshold voltage and reset threshold voltage are different for different devices. So the same voltage, because of identical pulses, may not be very much, I would say, may not be sufficient to induce set or reset in all the synapses in that network. For instance, if we have several of these RMs in a crossbar, same amount of voltage difference between the electrodes may not be sufficient to cause reset or set in other RMs. So what this leads to? This leads to these variations in the set and reset threshold voltage. They lead to different kind of STDP behavior, even with the same kind of pulses being applied in the across the two electrodes of the RM. Since the RMs have different set and reset threshold voltage across your array, crossbar array, so different pairs of RM will show different kind of STDP behavior. And these were the different kind of STDP behaviors which was shown by the different RMs in a crossbar because of the variations. And the work that Tejas did, we kind of modeled those variations, put that in a spiky neural network, and looked at how the effective performance of the neural network changes. And we had several very good conclusions, several interesting conclusions. If you are interested, you can go ahead and read this paper of Tejas. So with that, I would just finish this module. Here, I just wanted to give you a brief overview of what exactly are neurons, what exactly are synapses, what they do, what they perform, like how they do those things, and then what is the learning rule behind the unsupervised learning of these synapses and neurons, and how exactly we can implement these functions on hardware. Because unless we know about what neuron does, what synapse does, how we can model them electrically, how we can implement these learning rules, we won't be able to make any dedicated hardware which would kind of mimic those characteristics and achieve the energy efficiency levels of the human brain.